Right. Right. Good afternoon and happy Korean Thanksgiving to everyone. My name is Subin, and today I'll talk about our method, which is learning a genome-wide score of conservation at the functional genomics level between human and mouse. Our work is motivated by the fact that it's useful to know very often if a human locus is expected to have a similar role or functional properties to a mouse locus, especially when you have a set of variants or regulatory elements in human that you want to test in a mouse model, or when you want to map epigenetic signals from one species to the other. While pairwise sequence alignment allows us to map genomic regions between the two species pretty easily, there are many ways to infer whether those aligning regions will actually have similar functional properties. So one very good source of information is this growing collection of human and mouse functional genomics data that's now available. So we reasoned that it would be beneficial to use that data in order to highlight regions with strong evidence of conservation at the functional genomics level, and also provide complementary information to existing conservation annotations, such as sequence constraint annotations. We therefore developed our method called LECIF, which stands for Learning Evidence of Conservation from Integrated Functional Genomic Annotations. LECIF takes two large compendia of human and mouse functional genomic annotations as input, as shown on the top of the figure. And as a result, it learns a genome-wide annotation called LECIF score, which represents conservation at the functional genomics level. Unlike most comparative genomic methods, we try to take full advantage of available data by integrating different types of annotations, such as DNA-seq, ChIP-seq, and RNA-seq experiments, and we do so without explicitly matching them between human and mouse by their biological origin and type. Going to the method, we first define for each human or mouse region a feature vector that describes the functional genomic annotations overlapping those regions. So for example, in this human region A, there's a DNA-seq peak in liver and some RNA-seq signal in lung. To train, we first define our positive pair as regions that align to each other at the sequence level based on pairwise sequence alignment. Because they align to each other, these are likely to be similar at the functional genomics level. And as our negative pairs, we define them as regions that align not to each other, but somewhere else in the other species. And because they do not align to each other at the sequence level, they tend to be less similar than the positive pairs. And given these positive and negative pairs, we train an ensemble of neural networks, which allows us to make genome-wide prediction for regions that were held out from training. And we call the output of this classifier the LECIF score. And I'll just briefly note that we focus on aligning pairs of regions in this work, which is about 40% of the human genome, but the method can be applied to score any pair of human and mouse regions to quantify their conservation at the functional genomics level. In the feature vectors, we use DNA-seq peak calls, histone mark and transcription factor ChIP-seq peak calls, chromatin and chromatin state segmentation, capanosis gene expression peak calls, and RNA-seq signal. These all come from public consortia such as ENCODE, Roadmap, Mouse ENCODE, and Phantom5. And in total, we have about 11,000 features combined for human and mouse. Right, so here's what the LECIF score looks like in the genome browser. Here I have at the top a human DNA segment, and in the bottom the aligning mouse DNA segment. And I've highlighted chunks of DNA that align very closely to each other in black boxes. Our score is shown on the top with black vertical bars. In the bottom, I have chromatin states that were learned for both human and mouse, so the states are easily comparable. Chromatin states, they are states learned from chromat-HMM, which allows you to systematically annotate DNA elements based on combinatorial and spatial patterns of histone marks. So I have the legend of these states in the bottom. What I want you to notice here is on the right, we have a high scoring pair of regions. And when you compare the chromatin states in human and mouse, they indeed look pretty similar. Both of these human and mouse regions are annotated by red, orange, and blue states. In contrast, if you focus on the middle, the low scoring pair, there is species-specific pattern happening, specifically these yellow and orange states in human HepG2 cell type, And when you compare that to the mouse cell type liver, you do not see those yellow and orange states, which correspond to regions characterized by K4ME1 or 3. So this is an example where we observe a high LECIF score, and we indeed observe similarity in the chromatin states between human and mouse. And also, when we see a low score in a pair, we observe species-specific patterns in their chromatin states. We more systematically quantify this by manually matching human and mouse experiments, specifically K27 acetylation ChIP-seq experiments matched by tissue type. So this was manually done, not for our method, but for validation purposes. And for each of these pairs of experiments coming from human and mouse, we were able to define pairs of regions that have peak calls in both human and mouse, or those that have species-specific peak calls, and also those that have no peak calls at all. And we observe that for pairs where both human and mouse overlap a peak, so they share this signal in terms of K27 acetylation peak call in a specific tissue type, they tend to score higher than those that have species-specific patterns or those that have no signal at all. So one thing that I want to emphasize here is that the classifier that we trained did not have any information given explicitly in terms of which experiments are K27 acetylation, which experiments are coming from which tissue type. None of that metadata on the annotations were given. So LECIF is learning this biological correspondence between experiments on its own, just based on the data. And lastly, we looked at a particular cross-species study on differential methylation with respect to diabetic phenotypes. We did not use any methylation data in our method, so this is a good way to validate. This study by Mothop et al. Used mice to identify differentially methylated regions, or DMRs for short, associated with high-fat versus low-fat diet. They then mapped these mouse DMRs, or differentially methylated regions, to the human genome based on sequence alignment, and asked, are they also differentially methylated in lean versus obese patients? And so they were able to identify these conserved DMRs versus mouse-specific DMRs that could not be replicated in human. And we observed that for conserved DMRs, we tend to score those higher in comparison to the mouse-specific DMR. So this suggests that our score could be useful in interpreting this type of cross-species epigenetic experiments, and also be used to map these signals from one species to the other. In summary, we developed a method called LECIF, which learned a score of evidence for conservation between human and mouse for a compendium of functional genomic annotations. And the output of our method, LECIF score, highlights regions with strong evidence of conservation. And we believe that our score will be useful in many applications, especially in terms of translating results between human and mouse. With that, I'll thank my PI, Jason Ernst, our lab members for their discussion, and my 2019 QCBio Big Summer students, Trevor and Grace. Trevor is in this call, I believe. They extended this work to human and rat. So thanks to QCBio for giving us that opportunity, and also thank our funding sources. We also have a bioRxiv paper. So if you're interested, check that out, and I'll take any questions.